hopefully we're on YouTube as well. Okay. All right, so welcome everyone. It's my pleasure to host this first KNAW Biophysics NL meeting of 2021. Yeah, and we have a couple of exciting speakers. Hernan Garcia from the University of California, Berkeley, and Clélia de Mulatier from the University of Amsterdam. In addition, as discussion panelists, we're joined by Peter Rijn Tenwolde from AMO and Marvin Tannenbaum from the Hubrecht Institute. Okay, so our format, as our regular attendees will know, involves two relatively short talks, two half-an-hour slots. That's half an hour including questions. And then after that, we have a 1-hour tutorial, which is given by one of our two speakers. And today it will be given by Hernan. So yeah, and yeah, I think we have, a good couple of exciting topics today. So, in due of time, I think we should dive straight in. So Hernan, I'll just say a few words to introduce Hernan. Hernan did his PhD at Caltech with Rob Phillips, doing a lot of interesting theoretical work on analyzing gene expression using the tools of statistical physics, really. Olivia. Okay, maybe somebody can mute that person. Yeah, and I'm not sure, Hernan, actually, if you were already doing experiments with Rob, but after his time at Caltech, he moved to the lab of Thomas Gregor, where he got into, looking at transcriptional bursts in the developing fruit fly embryo. Very beautiful work. And in the last few years, he's become an assistant professor at the University of California at Berkeley, where he has built up a group doing a lot of exciting work. And as I can see from recent publications, even developing new experimental techniques. So yeah, I'm really excited to hear about your work, Hernan, and the floor is yours. The screen is yours.
All right, let me see. I think, there we go. I need to share screens. But there you go. I guess everybody can see it. So thanks again for having me here. It's a real honor and a pleasure to meet old friends and meet new ones. Yeah. So this I thought this would be like, I was told it's 15 minutes plus 15 minutes of questions. I'm very happy to stop along the way. I don't need to make it all the way to the end. So please stop me at any point. I'm going to try to start from the very beginning, because I was told also that maybe some people wouldn't be super on top of all some of the basic biology things. So I'll try, as I said, I'll try to start from the beginning, and I'm going to apologize to some people like Peter Rijn, who I think saw some version of this talk already, but maybe that means now that he'll be ready with more incisive questions as I move along. Anyway, once again, thank you for having me over. And the work I'm going to talk about today is the result of an effort spearheaded by Megan and Nick in my lab with the help of Emma, Simon, and Armando. But it's also the result of wonderful collaborations with the lab of Chris Wiggins and his undergrad Baje Galstian, who's actually, funny enough, going to be now a postdoc with Peter Rijn, and also my UC Berkeley colleagues Mike Eisen and Xavier Darzak. And as I said, just to start from the very beginning, as you know, you have 10 to the 14 cells in your body, and each one of those cells has the exact same DNA molecule. Okay, and fun fact, how much, I don't know if you guys have done the calculation of how much DNA you make over your lifetime, but it's 3 light years of DNA. So you should try that at some point. Anyway, but all these cells have the exact same DNA, and as you know, despite having the same information, they decide to become neurons, red blood cells, skin cells, whatever they want. And just as a reminder, these decisions are mediated by the so-called central dogma of molecular biology, where the idea is that you have, let me switch to the pointer, laser pointer, there you go. You have information encoding language in genes, and this Xerox machine, this RNA polymerase machine, makes a copy of the gene in the form of RNA that gets then captured by a ribosome that translates that into a protein that encodes for function. And think, for example, about hemoglobin carrying oxygen throughout your body. But one of the great insights is that some proteins are produced in some cells, some proteins are not produced in some cells. And the reason for this is that there are proteins, all the proteins are in charge of regulating transcription, where you have activators and repressors that can dictate or modulate the probability of finding the polymerase Xerox machine on the promoter. So the overall question that I've been excited about asking or curious about is, what happens when you take these cartoons seriously and try to turn it into a mathematical model? So what are the mathematical implications of this model? And we don't ask these questions in the context of humans, but we do so in the context of fruit flies. So how do you go from a single cell to a fully grown fruit fly? And you can see on the right, cells within an embryo, a fruit fly embryo, going through multiple rounds of division. And the cool thing is that even though at this point, all the cells look exactly the same, each one of them has already decided what they're going to be when they grow up. And you can see these decisions manifested by these massive rearrangements called gastrulation. Now, the reason each cell knows its identity is because they're expressing a unique combination of transcription factors. And we know that these transcription factors are organized in a cascade, where there's activators and repressors expressing shallow gradients that leads to other genes expressing broad domains, that leads to genes expressing 7 stripes, that gives you genes expressing 14 stripes, that ultimately segments the embryo and assigns each segment an identity. Are you going to be a leg, a wing, or so? Okay. We don't only know the molecular identities of these activators and repressors, we also know their regulatory connections. So this is an amazing state of the art to start asking the sorts of questions that we're interested in. And yet it's not enough, because just like this is not a pipe, this is not a fly. And what I mean by that is, if I change the names in this network and I give it to the world's experts in fruit flies, they would have a hard time telling me that this is a fly. You cannot calculate a fly out of this. And one of the reasons I'm going to try to convince you today, and also during the tutorial later on, is the reason you cannot do it is because we don't have information about space, time, or concentration in these network descriptions. So what we're trying to do in the lab is to endow each one of these arrows with molecular and quantitative information that allows us to reach a predictive understanding of developmental decision making. Okay, basically, could I predict the developmental phenotype from knowledge of the DNA sequence? Just to go into it in a little bit more detail, my dream is to be able to know the DNA sequence, the arrangement of binding sites for activators and repressors, and be able to predict the functional relationship, the input-output function that relates input concentration of transcription factors and output patterns of gene expression. Okay, but to do so, you got to break this apart a little bit. You got to know some phenomenology so you can actually know what's going on under the hood, so you can write theoretical models. And a few different things that you might worry about is asking, well, if I look at a picture like this, I'm thinking of the overall average concentration, let's say, in the nucleus of the transcription factor. But ultimately, what matters is that how many of those transcription factors are bound to the DNA in principle. I can also look at the overall output of the transcript of the gene, but these macroscopic patterns of gene expression are created by single cells regulating their transcriptional dynamics. So the point is that to understand how I go from input to output, I need to also understand what's happening at the binding level and what's happening at the single cell transcriptional level. So today, the talk is going to be divided in 2 little short parts that are very much inspired by this great article by Joel Cohen called Mathematics is Biology's Next Microscope, Only Better. Biology is Mathematics' Next Physics, Only Better. So throughout this talk, I'm going to kind of take this idea of microscopes. What can you learn about some biological phenomena by thinking that you couldn't learn by using even the fanciest super resolution microscopes? So I'm just pushing this idea of the microscopes to make the narrative a little bit more fun. But first, I'm going to introduce you to some microscopes that we developed to be able to figure out what is going on at individual promoters during transcription throughout development. And then we're going to use theoretical microscopes and actual light microscopes to figure out how is this transcriptional dynamics in individual promoters regulated by the binding and unbinding of transcription factors. And I'm going to leave you with more questions than answers, and some of them we're going to try to answer during the talk. So stay if you're curious, or we can also go into details during the talk, the question and answer period. So in particular, we're going to try to understand how this stripe of gene expression called stripe 2 of even skipped is created. Okay, and this stripe is created by 2 activators. The names don't matter. They're called Picard and Hunchback. They're initially expressed along the embryo and create a broad domain of activation. And later on, the repressors come on, and they restrict the transcription of Eve to like this little string, okay? Now, the thing to realize is that everything, pretty much everything we know about pattern formation comes from pictures like this one that measure the accumulation of RNA in the cytoplasm, which means that we have no information about what's going on with individual cells in order to realize this pattern of gene expression. So then the first question we wanted to ask is, okay, what are actually, just look at it, what are single cells actually doing in order to create this macroscopic pattern of gene expression? And to answer that question, we use some technology that was invented by Rob Singer and Edouard Bertrand, who's now in Montpellier, back in the 90s, and that we implemented in flies when I was in Thomas Gregor's lab at Princeton, and that it was also implemented in flies simultaneously by Natalie Dostani at the Curie Institute. And this is the so-called MS2 system. And the idea here is that we add a little sequence to the gene that we want to look at that when transcribed forms a loop. The loop is recognized by a binding protein fused to GFP such that every nucleus in your embryo will have a little spot of fluorescence, and the fluorescence of that spot will be proportional to how many polymerase molecules are actively transcribing the gene. This is what it looks like when you put it all together. And as you can, so what you see in red is the DNA, and in green, the RNA signal. And you see how initially there's a broad domain of activation, okay? And as development progresses, you see how that domain of activation, that green domain, is being restricted. I don't know if people can see me, actually. Can people see me? I think your video, Tom, is locked onto you because I'm gesticulating. Not that it matters too much. But
I think in Zoom, people can see you. I'm not sure about YouTube.
Okay. No. It's just on the lower right, I see. I guess maybe, yeah, no, you must be right.
We see you both on YouTube and on Zoom.
Okay.
Go ahead.
Okay, so you see me as I gesticulate. Okay. Again, not that it matters too much. Anyway, so you see the stripe here. And one thing to keep in mind is that what you're staring at is at an in vivo single molecule experiment, because it's reporting on the transcriptional decisions made by an individual promoter. Okay, so you can zoom into one of these little cells and plot the number of polymerase molecules as a function of time. And what you see is that these dynamics are described by these trapezoidal-like shapes. And so what do they mean? So what you need to remember is that this technique tells you how many polymerases are loaded onto the gene. But in principle, what transcription factors like to do is to regulate the rate at which you load the polymerases onto the gene. These two quantities are not the same, but they're related. And you can relate them by a simple model where you say that initially the promoter is in the off state. Then you turn it on and you start loading polymerases one by one, and that's why the fluorescence grows linearly. If the first polymerase, there's a question from Marvin. Hold on, I missed it.
Yeah, I was just curious, why do you only see one spot per nucleus instead of two?
Because we use heterozygote embryos for a reporter. The first version of this technology was with two, but then it got too tricky to track them. But actually now we're doing stuff where you can track both, and you can start asking questions about intrinsic and extrinsic noise, or correlated or uncorrelated noise. So there's fun stuff to be done there, but this makes the analysis a lot easier. Yeah, great question. Anyway, so I was saying, if the polymerase, if the promoter stays on for long enough, the first polymerase is going to fall off the gene. You're going to keep loading polymerases, which is going to lead to a uniform level of fluorescence. And finally, if you turn off the promoter, you're going to lose polymerases and fluorescence at the same rate that you loaded them. So the point I'm trying to make is that these trapezoids are consistent with a model where the promoter is on for just a little period of time. And these are these so-called transcriptional bursts. And people have seen them all over the place, in bacteria, in yeast, in mice, and we've seen them in zebrafish now. I mean, they seem to be the rule rather than the exception. And the simplest theoretical model onto which to project this phenomenology is the so-called two-state model, where what you posit is that the promoter can fluctuate stochastically between being in an off and an on state. Only when in the on state can it transcribe. And given this model, then you can define dynamic parameters such as the burst frequency, the burst length or burst duration, and the burst amplitude. Okay. So now that we know that the cells that are making the stripe are bursting, the question of how the cells make the macroscopic pattern becomes a little bit more refined, because the question is, how do you modulate these bursting parameters to create this mean rate of transcription that leads to the stripe? And when you go through the math, this is not too hard, at least in steady state. What you predict is that the accumulated RNA is given by a term that has to do with the fraction of time the promoter spends in the on state, and the rate of transcription when you're in the on state, which tells you that in order to make the stripe, you could do so by increasing K on, or the burst frequency in the middle of the stripe, decreasing K off, or increasing the burst duration in the middle of the stripe, or increasing the amplitude of the bursts in the middle of the stripe, or any combination thereof. Okay, so it doesn't have to be one of these three, it can be any combination. But regardless, if you want to answer this question, clearly what you need to do is be able to measure K on, K off, and R, the three bursting parameters in the embryo as you move along the embryo. And this would be trivial if you had access to the promoter. That is, if you had access to the promoter state, right? You would just measure K on, K off, and R, and you would be done. But the promoter state is hidden from your view. What you can see is the number of polymerase molecules that are loaded onto the gene. So you need to do some sort of inference. And this is easier said than done. I mean, we developed this technology in 2011, and it wasn't until 2018 or 2019 that we figured out how to do this inference together with Chris Wiggins by repurposing hidden Markov models. And I'm very happy to talk about some of the underlying math if you guys are curious about. But basically, the point is that now, after a little bit of hard work, we finally were able to analyze the terabytes and terabytes of data that we were sitting on. And so now we can do things such as this. Here I'm showing you the fluorescence signal, the number of polymerase molecules as a function of time for an individual nucleus. And you would have a hard time telling me when a burst begins and when a burst ends. But now we can find the most likely trajectory of an individual promoter using hidden Markov models. Not only that, we can do this for every nucleus throughout the embryo. So this is the same movie I showed you before, but now I'm color-coding the nuclei according to whether they're active or inactive. And what you can see is that even in the middle of the stripe, where transcription is supposed to be highest, you see lots of on and bursting. So bursting is there all the time, even at the maximum levels of gene expression. All right, so now that we have this tool, we can measure K on, K off, and R as you move along the embryo. Here I'm showing you the burst frequency, K on, as a function of position, and you see that sure enough, it's modulated in a stripe-like fashion, okay? Which tells you that some of the 4 transcription factors that activate even skip stripe 2 dictates the K on. When you look at burst duration, K off, there is no modulation, which tells you that none of these transcription factors touches that parameter. Interestingly, also the burst amplitude is controlled by some combination of these 4 transcription factors. And this is basically pretty much consistent with what everybody else has been seeing. Sometimes people see a control of burst frequency or burst duration, sometimes some combination of the two. Like there seems to be a whole zoo of these strategies, though there seems to be some unity, and I'll talk about that later on. But the main point is that we as a field have gotten pretty good at figuring out which bursting parameters are being regulated by transcription factors, but we're still lacking in terms of being able to answer how is this control implemented at the molecular level. Okay, let's see, Peter Ryan.
Yeah, I was just wondering whether it's K on itself that is modulated, or K on times the concentration, and then perhaps the concentration that is modulated.
Yeah. No, I think presumably, I mean, you can imagine a very simple model where if, let's say, only K on is being regulated, it's because when the activator binds, that's when you burst. So the frequency of binding is the frequency of bursting, which means that the concentration would upregulate the burst frequency. Absolutely. So I don't know yet, because as you move along the embryo, the 4 transcription factors are changing in concentration in all sorts of different ways. So what we need to do is to be able to visualize both inputs and outputs simultaneously. Okay, and part of that is actually now enabled with the technology that Jacques Bosma, who's now a colleague of Marvin, developed, that allows us to see those transcription factors in real time. And I'm very happy to talk about that. The one thing that you might worry about is that because you have 4 transcription factors, like looking at all of them simultaneously is kind of hard. So you might want to use a simpler system. But yeah, I think the idea is that K on is the simplest model is one of K on regulating the burst being related to binding, though I'll show some evidence against that in just a few slides.
Thanks.
Sure. All right. So I was saying, like we're really good. I mean, you give me any gene now, I run it through the hidden Markov model, or people do single molecule fish and do some theoretical modeling and figure out which bursting parameters are being regulated. But we still don't know how this control is implemented at the molecular level. And to do that, you need to go from actually looking at the single cells and, related to Peter Ryan's question, you need to be able to correlate something about the concentration of the transcription factors, their binding state, and the transcriptional activity. Okay. So in the second part of the short talk, I want to discuss how can you figure this out. And you can imagine like extreme scenarios, such as you can imagine that the transcription factor needs to be at the locus throughout the whole burst, but also maybe it just needs to be there at the beginning of the burst. And each one of these scenarios would have different implications for how bursting control is being realized. Okay, so it's a little bit tricky to measure both binding and transcription in the same locus at the same time, though we're making progress. But we decided to start with just putting on our theorist hats and seeing, what are the scenarios where we could make any sense of how transcription bursts are controlled by transcription factors. And the simplest model, and goes back to Peter Ryan's question, could be one where you have an activator binding site, and when the activator is found, that's when you're in the on state. Okay, so like this is the dumbest model I think that you can come up with for transcription factor control of bursting. And in this simple model, you make two predictions. One is that the burst frequency is equal to the activator binding rate. Another one is that the burst duration is equal to the activator residence time. And because burst frequency and activator binding rate are the same, burst frequency should be controlled by the activator concentration, whereas burst duration should not. It should be modulated if you can tune the binding affinity of the binding site, because that determines the residence time. But this makes a super simple question. It's super simple. It begs for a simple question, or some easy thing to do, which is just measure the binding and unbinding of these transcription factors in B1, see whether these predictions are matched. Okay. So to do that, we were very lucky that Xavier Daszak had built this fantastic lattice light sheet microscope, which I don't know how many of you are familiar with them, but by most measures I can think of, they're like 100 to 1,000 times better than confocal microscopes. And so previously people could measure binding and unbinding of transcription factors in single cells, but lattice light sheet really made it possible to do it in embryos. So what you see here is a field of view of nuclei in the embryo. There's a nucleus, another nucleus, another nucleus. And here you see a bicoid molecule. This is one of the activators of the stripe fused to GFP. And the reason you see now as a little fluorescent spot or bright punctum is because it's bound to the DNA, so it's staying there put for a little bit. If it's not bound, it moves too fast and you don't see it. Okay, so this is, I don't know how well this shows up, but hopefully in the movie you can see little flashes of fluorescence appearing, and those flashes correspond to binding events. Okay, and what you can do is then ask how long is each one of those events, how long does each one of those events live for, and plot a whole distribution of binding events. Okay, and what you see is that on average, bicoid stays for 1 second on the DNA. Now, this would be all good if it wasn't for the fact that the duration of the bursts that we're looking at is on the order of 5 minutes. Okay, so there is a disconnect between the two, between the binding phenomenon that we think has something to do with bursting and bursting itself. And this disconnect is kind of widespread for, I would say, for any molecular phenomenon that you think might be behind bursting. So in this little graph I'm showing you on the top, the bursting timescales that that people have measured in a bunch of different cells. On the bottom, I'm showing you the different timescales of molecular processes that you would say, well, maybe these guys are in charge of bursting. Okay, and what you see is that's not only binding, but many of the other phenomena are much faster than the timescales of bursting that we observe. Okay, so we're missing something. Okay, and I think that what we're missing, and this is something that we can discuss in the talk later on, is the fact that when I showed you the measurements of bicoid binding, when everybody measures binding, they don't know where they're measuring the binding. They're measuring the binding on the DNA. Sure enough, it might be specific binding, non-specific binding, but they don't know whether it's at a locus that is active. Okay, so maybe the binding is different when you look at a random location versus when you look at a target locus. Or maybe most of this binding is non-functional and just a small fraction of binding events are functional. And in order to know that, you need to know something about what's happening with the transcriptional state. Okay, so what I'm going to argue, like one of the things I would like to discuss in the talk later on is how do you go from the left to the right? How do you go from actually correlating binding events with the transcriptional activity? And the hope and the dream, and this is truly I don't know how many of you have had this experience. It's something that Rob and I talk a lot about, which is experiments that are worth changing your life for. And I've had a few of these in my life, and I think we're getting to the point where we're going to get to this experiment that will change my life, or at least will dramatically change the direction of my lab. Because my dream right now is to be able to measure, not only measure the progression of a transcriptional burst, but also to measure the binding and unbinding of activator during the burst, to measure, let's say, if a pioneer transcription factor opens a nucleosome, and also the dynamics of the, let's say, the chromatin landscape in a living embryo. Basically, what we're trying to do is at spatiotemporal resolutions that you cannot do with genome-wide technologies, to be able to do this in vivo biochemical dissection of what's happening in a burst, this anatomy of a burst, that I claim is necessary if you want to derive governing equations of cellular decision making in development. So with that, I'm just going to finish. I kind of gave you this idea of these microscopes that we can do computationally using Markov models. Like MS2 is I guess a molecular biology microscope. Lattice light sheet is an actual microscope. Theory is a microscope. But the point is that we're trying to put them together to see whether we can predict developmental phenotypes from the network structure. Okay, and I showed you an example of how to think about stripes, and it's tempting to think about the fact that maybe there's such a thing as a developmental basis set that you can, if you know how to learn how to make stripes, steps, and gradients, maybe you can one day figure out how to make an actual animal. Though there's a strong assumption here that you are actually allowed to decompose development in a modular fashion. So with that, once I already mentioned these guys earlier, and I also need to acknowledge my lab. It's a great group of people to do science with, and these funding sources, and everybody for your attention and your questions. And I'll be very happy to take more questions. Thank you.
Okay, wonderful. Thanks, Hernan, for a beautiful talk. So everybody in the audience, you're very welcome to add questions to the chat. But we'll start with questions from the panelists, Peter Ryan and Marvin. Yeah, sure, Marvin, please.
Yeah, so I was curious about the affinity of transcription for specific genes. I mean, I guess it makes a lot of sense that it's sequence specific, right? That it has sort of a broad consensus motive that is combined to N, N, G, A, T, but depending on the exact sequence, the affinity will be different probably for every locus. Mm-hmm. So do you think it's reasonable to expect to get a single affinity? And also when looking at your dwell time distribution, I mean, I didn't look at it very carefully, but it didn't look like an exponential. Maybe it does. I mean, it looks like it has quite a long tail to the right.
Yeah.
So what do you think that has significance, that those might reflect sort of the perfect consensus sequence binding events?
Yeah. So about affinity, I'm still confused about what affinity means in eukaryotic transcription factors. In bacteria, you got the like repressor that has like 21 base pairs. It's very clear how you can modulate transcription binding affinity. These guys tend to have like a 5 base pair, 6 base pair consensus sequence, right? Though people think that some of the flanking sequences maybe make a difference, maybe because of mechanic constraints or something like that. But it's not clear to me how much actually you can what the dynamic range of modulation is for binding affinities. Obviously this measurement averages over everything. In this measurement, you could actually see binding events that were specific and non-specific, and most people can see that as well, like in transcription factor. So there seem to be two populations now. They cannot distinguish within the specific population individual binding sites because they just don't know where things are binding. Right. So I think that's the first question. Hopefully that answered your first question. Now, the fact that this is looks a little bit off from exponential, I agree with you. And if you stare at the models, you could imagine scenarios where you predict that you have a very long tail. The problem here is that GFP lasts only 5 seconds or 10 seconds or something like that. So you're limited by the photon budget you have. Okay, so you might be able to get away with halo tags and all the things that give you maybe 10 times more photons, but maybe you need to do the types of fun sun tag thing, something that allows you to renew the GFP. So this is something that we've been excited to try, though we haven't gotten around to doing it yet.
Just a quick follow-up. I was wondering if you think about these essays. I forget who's doing this, where they do like huge, complex, diverse libraries, and then do single molecule imaging, and then sequence the libraries to see sort of the off rates and the on rates on, thousands or tens of thousands of different sequences. Do you think that's, that's going to be valuable in addressing these questions?
Like in vitro, like Mitomi, like the thing that Sebastian Merkel does?
Maybe.
Or in vivo?
In vitro.
But
so you synthesize oligos and then, combine imaging with sequencing.
Yeah, so no, we haven't thought about it. I'm kind of trying to figure out, like my focus has been mostly on the in vivo side, trying to see what we can learn from in vivo. I've always wondered, like, this goes back to my work on lac repressor, you can infer lac repressor binding energies, but it's hard to relate them to what's happening in vitro. So yes, I think it would be fun to do that. And people have done some experiments like that on Bicoid, though I don't think they've done binding dynamics for Bicoid. They've done select stuff on binding affinity. So that would be fun to do. But I mean, my dream would be to do the stuff that Johan Elf does in here, like actually see them at an individual locus.
Because I guess it would be nice. I mean, if you could show that there's no sequence, in the world that it has a residence time of more than 5 seconds on, then, that would be additional support for the hypothesis that it's, that it's not just a very long tail here that you're, not observing because of bleaching rates or something. It simply can, there's no sequence that will bind to more than for 3 seconds. And then,
I understand what you're saying. Yeah. And in fact, what we think is happening is that cooperativity is stabilizing some of the binding, that it's not the binding and unbinding of individual sequences. Yeah.
Okay.
All right. Thanks, Marvin.
I see Peter Ryan has already put a question in the chat. Would you like to?
Yeah, right. There's this interesting question still in the field about the precision of these expression domains, right? And there's this idea that temporal averaging would not suffice, and that spatial averaging would be required. And I was just wondering, you have these beautiful movies where you see these cells, transcription in these cells, and I was just wondering whether there's any spatial correlations in these patterns of transcription which may hint at spatial averaging.
Yeah, there's for sure. Let me see if I have, because maybe I have it here. Too many slides. But there's some cool stuff that Jacques did. No, I don't see it. I don't have it here. But yes, for sure there's sharing of stuff throughout the cytoplasm. I mean, this is something very, very specific, obviously, to the fruit fly, and we have examples, for example, of a cell that how does this work? A cell that has bursts of transcription surrounded by cells that do not express at all, and you see bursts of proteins appearing in this neighboring cells related to the burst of transcription in the cell in the middle.
Yeah.
So for sure, there's spatial averaging.
Yeah.
Of protein, I don't know how much RNA is, because RNA diffuses a lot slower. But for sure, proteins do diffuse, and it's super interesting because, like you get all these sharp patterns. So how do you reconcile that with protein diffusion? And so if you look at a lot of network models, like the ones that Jörg Jäger have been doing, or Gerta Böd, sometimes they have to ignore diffusion because the models get too complicated. But I would love to understand exactly what's happening with the cytoplasmic sharing.
Absolutely, I think that's very interesting indeed. It's great to see that. It's great to hear. So basically, indeed, so this is precisely this picture. You have a burst of synthesis, and then these proteins are rapidly distributed over neighboring cells, and that's really key to spatial averaging. That's the whole idea of it.
Exactly.
Yeah.
So
Okay.
let's see if I can. Yeah.
In the interest of time, I'm going to suggest we just make it one last question from the audience that I'll read out. For the rest of you that have more questions, of course, Hernan is still going to give the tutorial after the next talk. So there's plenty of time to ask questions afterwards. I'm just going to relay one question from the chat from Tineke Lenstra, who said, as you know, we have correlated activator binding with bursting in budding yeast, where we indeed found a clear correlation. Bursting timescales are different in budding yeast than Drosophila, but could you speculate why they may be mechanistically different?
I have no clue. And I was actually just reading your paper this morning, Tineke, and trying to understand, because I'm also we're working on a new book on transcription, and I'm in this chapter right now, and I was trying to figure out how to reconcile the whole thing. So yeah, Tineke has beautiful measurements of gal binding correlated to bursting, and I guess the bursts are much shorter, right? That's what you're saying, Tineke, and if I remember correctly. And I don't know how to reconcile them just yet. Yeah, sorry, but I would love. I mean, if you come over later, maybe you and I can chat with the whole crew and figure it out.
Yeah, so more time during the tutorial. So thanks again, Hernan, for a wonderful talk. It's really exciting to see these latest beautiful results. So yeah, I guess we could, if you could stop sharing your screen. Clélia Delatier is our next speaker. So Clélia has just arrived in Amsterdam from Pennsylvania at UPenn. So Clélia did her PhD in statistical physics in Paris, working on, among other things, sort of branching random walk processes. And from 2015 to 2017, she was a postdoc at the ICTP in Italy, where she was, I think, started her work in sort of biophysics in the quantitative life sciences group. From 2017 to 2020, she was a postdoc at UPenn in the physics department and at the Computational Neuroscience Initiative there, working with Vijay Balasubramaniam. So she's just arrived, as I said, in Amsterdam as an assistant professor in physics at the University of Amsterdam. So today she's going to tell us about searching for high order patterns of data, something I think a lot of us are interested in across very different branches of biophysics. So yeah, we're excited to hear about it. Clélia, the floor is yours.
Okay, that's good. Do you hear me actually? Because I can't see if my microphone is on. Do you hear me?
We can hear you.
Yeah.
Oh, okay, great. Okay. Okay, so yeah, so I'm going to talk about searching for higher order patterns of data, and in particular, why I think simple models are important in that context. And so let's start. So it's a very common problem to, you make an experiment and you measure some quantities on your experiments, so you observe some properties of your system, and what you want to do is to come up with a good model for that system. And so this is an inverse problem or a modeling problem. And so you have some noisy data and you want to find a model that best captures the patterns that are hidden in the data. And in general, I would say that it is common to, let's say you measure a certain number of variables, and it is too common to consider at best interaction between pairs of variables to model the system. And this has been pretty successful until now, but there are recent experiments and theoretical studies that show the importance of higher order interaction as well in various contexts. So I've given here different examples in the context of neural coding, and you see those are 4 papers that are very recent. And I would say that in general, we tend to avoid higher order interaction because it adds to the difficulty of the modeling problem, and also because they are generally difficult to interpret. So the question we would like to ask is that, you measure some certain data set and you want to know if you can figure out if there are some higher order pattern hidden in that system. And also if you can identify which one they are precisely. So those are, I would say, big questions. And I'm not necessarily going to give you an answer, precise answer to those questions, but I will tell you what is the path that I've taken to try maybe to reach an answer at the end. And so I will mainly consider stationary system because I think it's a little bit simpler initially. And actually, I've mainly focused on binary data. So these are different examples of binary data. For each of the data here, I gave you, so I don't know what is the background, but I guess binary data here are composed of a set of variables, of binary variables. And so, for example, if I take a certain number of voters, then the binary variable will be each of the voter, and the data set will be composed of the answers of the voters on different cases, let's say yes or no. But you can also consider neural data. in which each variable will be a neuron, and the binary data will be composed of, let's say, 1 and 0s if the neuron is spiking or not in a given time being. And you can come up with many examples of binary data. And so the common models for this type of data are spin models. So if I take an example that some of you knows very well, which is the US Supreme Court's data sets. So this is under the second Rehnquist courts. There are 9 justices in the US Supreme Courts. And this data set is composed of 895 votes. I took it from this paper. And for each of the votes, the justices that said yes or in favor or against the case, but the votes are labeled if they were conservative or liberal, if the answer is conservative or liberal. So let's say for each of the votes, the justices give a +1 if it is conservative and a -1 if the answer is liberal. And so this can be considered as a spin variable, let's say. And it is common to model this type of problem with pairwise models. So this is also known as Ising model. So for that, you have to assume that the system is stationary. And then you assume that each of the votes of the system are independently sampled from that probability distribution. So this probability distribution is parameterized by a set of parameters that are in green here. So the G vector is composed of the HI parameter and the JIJ parameter. And so this parameter HI model an external field that drive the voter to vote conservative if HI is positive or liberal if HI is negative. And the JIJ here are couplings that model the tendency of, let's say, justices I and J to vote similarly if JIJ is positive and oppositely if JIJ is negative. So you see in that system, the thing is that we have no notion of locality. So we don't know, maybe these justices tend to be voting similarly to these other justices, but we have no notion of who is more likely to be connected to who. And so a priori, we have to connect everyone. But we can also consider any other model which is composed of a subset of those pairwise interactions. Or even we could remove some of the field here. But we could also consider models with higher type of interactions. So a way to expand the Ising model that you saw earlier to higher order interaction is the following way. So those are known as spin models. And so I would just add, you see, for example, here I have added a 4-body interaction and here a 3-body interaction. And this 3-body interaction here, for example, will be represented in the exponential with a term of the form J457, which will be a parameter, and then an operator, which will be S4, S5, S7. And that will be, for example, here a 4-body interactions in the Hamiltonian. And so just to give you some intuition of what that means, the JIJK then will model a tendency of justices I, J, and K to vote according to the following pattern. So either 1, 1, 1 or -1, -1, 1 if JIJK is positive. So let's say either they all vote the same way and they all vote conservative, or if 2 of them vote liberal, then the third one will vote conservative. In terms of neurons, that would sound like, some type of inhibitory patterns. If 2 of them fires, then another one is always silent. Okay, so this extension of pairwise spin model to include higher order interaction is actually rigorous, and you can map any possible type of patterns for binary data using this expansion. Okay, so but what I want to highlight here is that by choosing the form of your expansion here, by choosing the form in the exponential, you're deciding already what are the relevant patterns in your system. So for example, if I choose this type of pairwise model, then when I infer the parameter, so I find the parameters J star that maximizes the probability to reproduce a data set, I will find that at the maximum, I have an exact equality between the average value of the SI in the model, the fit-in model, and the average value of the SI in the data, so the empirical averages. And the same for the correlation. So basically, when I choose this type of model, I decide that the relevant observable in my system are the average probability of the justices to vote conservative and the correlations between the justices. Okay. And so the observable that I consider relevant then would be exactly equal or almost equal at best fit between the model and the data. And then if I take my other observable, they will end up somewhere in that graph. And if my model is good to capture all the pattern of the data, then all this observable here will be somehow in a certain standard deviation from that x equal to y line, where the standard deviation is proportional to 1 over square root of n. Okay, so if this is the case, then I would say that my model is good at capturing the pattern of my data, Okay, so for example, if I take the fully connected pairwise model for the US Supreme Court, then these are all the observable of the system, including higher order observables. And what you can see is that they align pretty well along the x equal to y axis at best fit. Okay, but I could have actually taken another type of model with also 45 interactions. So the fully connected pairwise model has 45 interactions. And so I can take another type of models. And this one can include higher order interaction. And actually can take many types of models that kind of fit pretty well. And so the consequence of this is that there is what we can see here is that the degeneracy of models that fit similarly. And in particular, the fact that a model reproduces well all the pattern of the data doesn't necessarily tell you that the model captures something important about the relevant pattern of the system. Okay, and so the question you can ask is that, can I find another model, for example, with least number of interactions, but that still capture very well the pattern of the system? And so, for example, here I took a model with 15 interactions only, and that includes some type of higher interactions, and that still seems to model pretty well the system. So what you can say here is that maybe this 15 interaction will tell us something more about the system, about the underlying structure of the system. Okay, so the conclusion here is that it is important, the way we choose the model is very important, because by choosing this model, we are already making an assumption on what is relevant and what is not relevant in the system. But the fact is that we actually don't know what are the relevant patterns of the system. And so one thing that we have to understand is that selecting the model appropriately is very important if you want to be able to say something about the underlying structure of the system. Okay, I don't know if this is clear. It's a bit...
If you don't mind me asking a question,
yes, sure.
It might be a stupid question. You said in this slide that what you've shown before this basically, right, suggests that it's important what model you choose, but you showed very contrasting models that actually seem to fit the data equally well. So I'm a little confused.
Exactly. So the reason why I showed this, what I want to say here is that you can actually, because the space of model is so complex, is so large, you can come up with many models that fit very well the data.
Yeah.
So you have to find, if you want to say something from the model you have chosen and say something about your system from that model, you have to be sure that the model you have chosen is not just fitting very well your data.
Yeah,
you have to say something more about it.
You have to be mindful of the degeneracy, effectively.
Sorry?
You have to be mindful of the degeneracy that, I guess, that's inherent in this kind of modeling these complicated
Exactly. So for example, here, the fact that I find another model with least number of interactions that still capture very well the data, I'm thinking, okay, maybe this is not the best model, but probably will tell me something more than the model with the 45 interactions, but that was giving me the same thing. Okay, so the point where I want to go is that you have to properly select the model, and that's come up with this balance between fitting and simplicity. And so this is what I want to talk about now. And so that's yeah, sorry.
Oh, that's very clear. Thank you.
So and so, you know, when you start with taking all the space of all possible pairwise, all possible models for spin models for binary data, then you start with a model with 2 to the n models, which is an exponentially increasing number of models with a number of variables. And so how do we choose the best model? So what we have to do is what we just said, is we have to find a good balance between simplicity and goodness of fit. So we want a model that is simple that is not too simple, because we still want this model to fit the data. And we want also a model that is not too complex, because we want this model to capture the main pattern of the data, and this main pattern only, and not all the noise that comes with it. Okay, and so how do we do this balance between simplicity and goodness of fit? So there are two different ways of doing that. There is something called the Bayesian approach, which is basically take all the models that you have, all the model or your possible candidates, and then compute the probabilities that these candidates reproduce your data, whatever the value of the parameter. So this quantity is integrated over all the value of the parameters that you can have in your system, given a certain choice of the prior of the parameters. The second option is some type of information theoretic approach. And so there are many different, you have probably heard about Bayesian information criterion or minimal description length principle. And so the idea of this approach is to balance between the goodness of fit and the complexity of the model. So what you want to do here is to find the model that minimizes this quantity. And what is this quantity? The first one is minus the maximum log likelihood of the model. So the better the model fits, the larger is the maximum log likelihood, and so the smaller is that quantity, okay? But there are many models that can fit the same way, we just saw that, and so what we have to compare is how complex they are. So the complexity of the model is something that tells you how flexible is the model to fit broad type of data. So the model that is very complex will fit various type of data, and so in general, they will have a very good fit for many type of data. So you want to balance these two things. And so I will tell you a little bit more about this model complexity. So in particular, in case of spin models, you can decompose this model complexity in two terms. So this is what is written here. The first term is proportional to the number of parameters in your system. And it's case the number of parameters. So the more the number of parameters, the more complex is your model. The second term is this geometrical complexity. So the geometrical complexity is actually in general very difficult to compute. What I can tell you is that it does not depend on the order of the interaction. So you cannot say, okay, I'm going to take only pairwise model because they are simpler. So they look simpler, but they're actually not simpler in terms of model complexity. And what we show is that the model complexity does not depend on the order of the interaction, but depend on the topology of the model. And the more compact in the model, so if you take all these spin models, if you put all your interaction in a compact way, the more compact is the model, the simpler will be the model. And so in particular, a fully connected pairwise model that is tend to be very expanded tends to be also very complex and will fit a lot of type of data. Okay, so the second thing I want to tell you about this expansion is that the first term grows with n. So let's say that n is the number of data points that you have. The first term, which is the maximum log likelihood, grows with n. The second term grows with log n, and the second term is of the order of 1. So if you have a very, very long data set, then you might not really care about this complexity term, and you only care about the fitting. But now, if your data set is actually finite, which is the case for almost everybody, you will start to have to care about these other terms. Okay, so if we come back to our initial problem, we have this giant pool of 2 to the 2 to the n model. So if we have the 9 justices, that's 10 to the 153 models, that's enormous. And we want to use one of these techniques, and we have this problem that in general, all these terms are complicated. So this Bayesian model selection is complicated because you have to integrate over all the parameters. And the minimum description length principle is complicated because the fitting is computationally demanding. So if you have to do it for all the data, all the model is complex, and computing the complexity of the model is very hard. So in general, this is very complicated. But we have tried the simplest approach to start from exploring this giant space of model and try to see if we can come up with a way to find what are the main relevant patterns of the system. So what we did is that we start with no interaction at all in our model, and we add interaction one by trying to figure out what is the next most important pattern of the system. So we will try to find what is the observable that is the most biased in the system that seems to be very important. We add it in the model, we refit the model, and we try to see what is the next observable that is the least captured by the current model. And so that's some type of greedy procedure. These are our data. We start from 0, and we try to draw a path from 0 to the data. And so this is feasible numerically until 15 to 20 variables. But the same problem arises is that we can find that there is a giant generacy of models that will perform similarly. And the smallest in the data sets, the larger is that degeneracy. And the largest is also the number of variables, also the larger is that degeneracy. So this is a big problem. And so I think one of the main questions that we haven't asked, and one of the main problems that we have to solve first, is the fact that we have no notion of locality in that system. So if we were to have a notion of locality, then we will not have this giant pool of model, and we will be able to restrict the type of model we have to consider. Okay, and so this is my last part. And so that's how to come up with that type of notion of locality in these complex systems. And so we have called this minimally complex model because the model that we will use here, they tend to have low complexity. So, okay, so I don't know if I've lost everybody or not. So maybe, okay. I'm going to go quickly through this part. It's actually a bit short. So this type of minimally complex model, so instead of considering model selection among all possible pairwise models, as most people are doing, now we are proposing to do a model selection among different type of model. And these models, they take this form. So let's say that these points here, they are random variables. And what you consider is that you... sorry, I said someone is asking a question. Okay, I think someone has this microphone on. But yeah. Okay, so instead of considering, so we're considering this type of models where you take, let's say, for example, here I have 6 variables, and I will take all possible interaction on a subset of variable. So for example, I take 2 subsets here, those 3, and those 3, and on each of the subset, I put all possible interactions. So all the fields, all the pairwise, and the triplets. And this is the same here. And this is what we call a minimally complex model. I can do another example here. So this one, I take the 6 variables we had initially, and I separate them in 2 packages. One package has 2 variables, and the other one has 4 variables. And on these 2 variables, I put all the interactions. So that's 2 fields and 1 pairwise. And on these 4 variables, I also put all the interaction, which has all the fields, all the pairwise, all the triplets, and all the and 1 4-body interactions. Okay, so this is what we call minimally complex model, are all the model of this form. Okay, so not only they're all the model of this form, but they also all the model of this form in any possible basis. So I can take my data, I can transform it in another basis, and I can look at model of this form in this other possible basis. Okay, so what I want you to tell here is that this type of models, the model selection using this type of model will not depend on the basis in which you're looking at your data, which is also interesting. Okay, so why do we like this type of model? So first, there are less models. Instead of 10 to the 153 here, we will start with 10 to the 20. Then they are simple in terms of model complexity. So this information theoretic model complexity I showed you earlier. So this type of model are simple. And then the last thing is that they have an easy interpretation. So we saw that 3-body interaction and body interaction earlier were already hard to interpret. There is this combinatorial notion. 4-body interaction is even harder. But these type of models are easy to interpret because they tell you about dependencies and independencies in your system. They will tell you about communities in your system. And somehow they will inform you about notion of locality in your system. So if you're able to find out a good model of this form for your data, then it will tell you how locals are your variables. And that's already a good thing. And the next reason why we like them is because all these quantities are very easy to compute. So it's easy to compare models in that context. So what is the algorithm to do that? So let's take our US Supreme Court data we had earlier. Then the first thing we have to do is to find a basis in which the data set is closest to being independent. And so for the independence for the US Supreme Court, that basis is the following. It looks like that. So this is our there are 8 pairwise interactions. So I have represented all of them here, and there is 1 field. So that's a basis, actually. I haven't explained you what this is exactly, but you can take your data and transform them in that basis. So my initial data, if I compare all the observable in the model versus all the observable in the data, for an independent model in the initial basis that looks like that. It's pretty bad. But if I go in my new basis, which has only 9 interactions, it looks like that, which is already really good. And those 9 interactions alone, they capture 86% of the information captured by the pairwise model, by the fully connected pairwise model we saw earlier. So we can say that somehow those interactions, they tell us something about the system. Okay, and then the next thing we have to do is we transform the data in this new basis. So now I have new basis components. So they have 9 variables here. That was this pairwise interaction we saw earlier. And in this new basis, I will look for the minimally complex model. So the packages, the combination of packages that will fit the best the data. And so it looks like that. So there are 3 packages. And if I pull it back in the US Supreme Court, how does it look like? It shows me that there is one package that happens to be the packages of the liberal justices, and then another package that tends to be the package of the most of the conservative justices. And then there is this pairwise interaction between the 2 most conservative justices that is very important and tend to be independent from the rest of the system. Okay, so about this minimally complex model, we like them because they will tell us, will give us a notion of locality in the system, and they also give us some type of, they tell us also about some orders of relevance of the dimension of the system. So this I haven't exactly tell you about it. And they allow to do dimensionality reduction, and so on and so forth, and they don't depend on the basis in which we have looked at our data. And there are other computational reasons why they are interesting. Okay, so but what's next? And so now that ideally we have an enormous number of variables, we record an enormous number of variables, and what we want to do is to find out notion of locality. So assume that we're using these spin models to do some notion of locality. So that makes way simpler. Now we can look inside each small communities, and in the small communities we can look for interactions. And because we have a smaller number of variables, we can use some type of greedy algorithm, or we can use some, we will have less degeneracy of models in the smallest number of variables, to model this smallest number of variables. And the idea might be then to do some type of coarse graining and to keep on like this. And so, okay, that's my last point. We have started to do some comparison, to support these ideas. Comparing different methods. And so the first one is the statistical method using PCA, ICA. Sorry, maybe this is a bit long, but assume that you have some neuronal data. We have used PCA and ICA technique to compute assemblies, to do assembly detection on this data. And this is a known technique. And so, for example, in this data, we found 2 assemblies, one on neuron 6, 8, 9, 10, 11, and the other one, this is orange assemblies. And then on the same data set, we can run a pairwise model selection, for example, and we will find this type of pairwise model selection, this type of pairwise model, and run a community detection on this pairwise structure, on this network. And if we do that, we find something, we find similar type of communities. You see the orange communities is kind of similar to this one, the blue community is similar to this one. And now on this same data set, we can also run this analysis that I've performed, which is minimally complex model analysis, to find, communities. The difference is that the 2 first case 1 and A and B, they only assume pairwise interactions between the variables, whereas the last one also allow for higher order interactions. So interestingly, we find similar communities. So community structures are very stable, but we also, find this type of 3-body interaction that is very important here, and that allow this neuron 6 to be stably placed in the blue communities. Okay, so I don't know, this is I went very, very fast. So please ask me questions, and if there is time. And okay, so yeah, just to tell you, this is a very long stories. I try to put in very short slides, thoughts that came up with, that we discussed with all these people here. And yeah, so yeah, please ask questions.
Okay, thank you, Clelia. A very interesting talk. I'm sure there are many questions. As usual, I'll give the panelists sort of a first shot. Who would like to start? Peter Ryan, would you like to go first?
Yeah, I could start. Right, so you mentioned at the beginning that one of the main assumptions is stationarity. But I guess many interesting problems are indeed non-stationary. And so how much progress can you make to extending these techniques to non-stationary problems?
Yeah, so there is one thing that I find interesting is that this type of community structure, using this type of models, they tend to be very stable. So even if your system is non-stationary, I think there is an interesting thing that can be done, which would be to assume, assuming that the system is stationary, even though it is not the case, might still allow to find some, how to say, the community structure of the data, which is some type of overall community structure, if there exists. So, for example, even if your system is not stationary, but there is still this tendency that, let's say, the 3 neurons or 3 variables tend to interact a lot with each other over the whole recording, you might still, by assuming the system stationary and using this type of technique, find out that, you might still find out the community structure, and that might still be useful later on to use in your non-stationary model. So that's one thing. And then the other thing is that I think that it might be that in the non-stationary case, it might be easier to find out if the system has high order pattern or not. I don't know. So I haven't studied, I haven't studied any, I haven't tried to make any progress on the non-stationary question. But there might be more clues that in your non-stationary system, there might be more clues of high order patterns. The clues might be easier to see than if you consider system stationary.
Yeah, thanks.
Okay. Yeah. Marvin, want to have a go? You put something in. Oh, sorry. I see you wrote something, but I think you were maybe.
I think probably there's other people that have more interesting questions than me for this topic.
Okay.
So I have a question for Clelia. Clelia, so I'm curious to what extent you're limited by data to assess the quality of higher order models. So for instance, if you look at the example of the US Supreme Court, the number of cases that you can actually assess is limited. I think it was about 1,000 or so. To what extent does this make it difficult to assess the quality of models with increasingly higher order complexity? In other words, the error bars on the higher order interactions become larger and larger, I guess, with higher order. And so how do you deal with that? Yeah,
so it is not true. So in the case of spin models, so if you consider this spin model with -1,1 convention, the higher order or the lower order interactions, they are completely equivalent. So I haven't gone through that part, I skipped it, but you can show that it doesn't matter if you consider 2-body or 3-body interactions, and the error bars are the same. So I can take a model with pairwise interaction, and I can I can generate data from that model, and then let's say take the data, and you choose a bijection. You look at your data in another basis, and you can make a bijection that will map your data in a new basis in which the binary, your initial pairwise model is now a model with high order interactions. So in that sense, the error bars on your high order patterns, they are not increasing. That is not correct for this -1,1 convention. So it is true in this 0,1 convention, there is a subtlety here. But I'm not sure that if you extend this 0,1 convention to higher order pattern, I'm not sure then you will map an entire basis for binary data. So this I don't know. I can check this, but I'm not sure. So this is one part of your to answer your question. But the second thing is that indeed, if you have a finite data, if you have a very small data set, you will have a huge degeneracy of models that fit similarly, and this is what we saw earlier. And the smaller is your data set, or the more noisy at the same time is your data set, and the bigger is that degeneracy. And so that makes it harder to really say if you have high order patterns. And so unless obviously, if you only use pairwise interaction and you see you don't reproduce the pattern of your system, this is, in that case, that would be clear that you're missing something. And this is the reason why I think it is important to reduce the space of models. And one way to reduce the space of model is really to look for this notion of locality. So if you have a notion of localities, then you can really simplify your problem. And then you can look for a submodel in each subsystem. And on a smaller set of variables, then you need least number of data points to be good. So yeah, so I think this is how I would answer your questions. Yeah.
Thanks a lot for clarifying.
Thanks. There are a couple of other questions in the chat, but we're also running a little bit behind schedule. So I'm going to maybe suggest that if Clelia has time to answer those questions over the chat while we move on to the tutorial, that would be wonderful. But just using my chair's prerogative, I wanted to ask one last question because I did find your talk very interesting. You know, I think it's really exciting that you're kind of exploring how to go beyond these pairwise spin models that are already very powerful, I think, in explaining all kinds of data. But I wondered, with your approach, you know, one question I wondered is, you know, if you, just as a kind of sanity check, if you generated data using a pairwise interaction only model, does your approach correctly come to the conclusion that the minimally complex model is a pairwise model? So that's one question. The other part of the question is, I'm wondering if there's like a real world example where it's very obviously necessary to, you know, consider more than pairwise interactions. I mean, you have the Supreme Court example, which is a very complex system, but maybe I thought if there's a bit of a simpler system that everybody can understand, that everybody can obviously kind of understand that you need more than pairwise, but is maybe, you know, something that you could easily demonstrate the power of your approach on.
Yeah, so I should have given this nicer example actually. So to answer the first question, so the minimally complex model thing is not. I mean, it will not tell you this has to be pairwise. It will tell you this is how variables are connected, more likely to be connected to each other or not. So for example, if you start with a pairwise model, you generate data, and then you run the minimally complex model selection, then it will map out how, sorry, let's say you start with your pairwise model and you place your interaction in a certain way. So not uniformly, but in a certain community way, or you might have connection between communities as well. Then this minimally complex model will highlight the communities. And then it's up to you to keep on, the procedure is not finished. So now you have a notion of locality, and from that notion of locality, then the idea will be to look for interaction within each communities, and then later on between communities. So I haven't, it's a good idea, I should actually do it. I haven't done the question that you're asking, and yeah. I think it's a very interesting thing to do.
I think it would be interesting to see what comes out, yeah, okay.
And then just quickly, the second question, it's just an example, and I do have one that I find interesting. This one is an example on songbirds. So we had the data recorded. I don't know. I don't have the name here, but I'm working with I'm starting working with Mark Schmidt at Penn, and we have some data of like here it's a community of 16 birds that are singing to each other. I mean, they are interacting with each other. They are social birds, and the data I had is only the singing part. And so I also have the mating patterns of the birds, and just using the singing, so singing and vocalization. So there is the male are singing and the females are vocalizing, are replying to the male. And so what you can do is that you can binarize your data sets by just using some time beans and then say in each time bean is vocalizing or not. And using this binary data, then you can use this minimally complex model selection and you will find out these communities. So they are, let's say, 4 big communities, and then some birds that tend to be kind of out of the system. And interestingly, in this community, there are some very important 3-body patterns. And these 3-body patterns are generally between 1 male and 2 female. And if you look back in the data, what you will see is that there is one pattern that is very recurrent in the data set, which is when one male sings, one of the female is singing, but the other one is always silent. So it will be, you will see this 110 or 110 as a very high frequency in your system. And it's more important than some pairwise pattern in the system. So this is one example.
So you can recover the love triangle.
Exactly, you can recover the love triangle.
Nice. Okay, wonderful. So thanks again, Clelia, for a very interesting talk. So again, yeah, if people, perhaps you could answer some of the questions that are already in the chat. And yeah, I encourage other people to continue asking questions in the chat as we move on to the tutorial by Hernan Garcia. So Hernan.
All right.
It's
so.
Let's see. Okay. Yeah, the screen
Let's
is.
see.
Yeah. Yeah, we are.
Yeah, actually, Clea, I would love.
Let's keep going.
yeah, Clea. Yeah, I mean, but I'm so curious. What the, going back to molecular implementation. What's the actual implementation of interactions? Like how do they in the Supreme Court? I'm just curious. What does it mean to have higher order things? Like, do they talk about things, or I'm assuming they make decisions by themselves? So that I was amused by to think about that. So Tom, how long do you want me to go?
So officially, I guess the scheduled time would be 45 minutes from now. Officially, we had allocated an hour, so anytime.
That's fine. That's fine.
That range where you're comfortable. I think.
Yeah. I mean, I think we can be done within 45 minutes, especially given that I think most people that are here were also here beforehand. So
yeah.
So So
okay.
Go ahead.
Yeah. So yeah, anywhere within
okay.
45 minutes to an hour will be.
Sounds great. Sounds great. So yeah. So I mean, I guess I was told this is supposed to be even more informal than the first one. So just interrupt me at will, and maybe what I'll do is I'll go over some of the slides that I showed you already, but then try to go a little bit more in detail, and also try to emphasize the challenges and problems and ways that we think we might have some way to address them, but in many cases we're still kind of hopeless. So any ideas are welcome. Okay, so let's see, it's working. Yeah, there you go. So I talked already. I introduced you to the crew, and once again, just remember that we have this amazing state of the art where we know the network of the transcription factors that dictate cellular decision making, at least along this long axis of the embryo. Meaning we know the molecular players, we know their interactions, and yet, as I said, if I gave you this network, it's very useful, but you kind of calculate a fly out of this, and that's kind of like my dream. Could I give you a DNA sequence? Could I tell you what the binding sites for activators and repressors are, and could you figure out what the cell is going to be when it grows up? Okay, so to put this in other words, could you predict input-output functions? Just to make it a little more specific, like imagine you have an electronic circuit. I draw you the circuit diagram. You open one of the Horowitz and Hill book, and you can predict precisely what the output current is as a function of the input voltage of that circuit. Nobody can do that if I give you a DNA sequence. And forget about a DNA sequence, if I tell you what the binding sites are for each transcription factor. So can theory and experiment agree on these input-output functions, on the functional relationship between output level of gene expression and input transcription factor concentration? And of course, a fair reaction that you might have is like, this guy's crazy. Like, how can you have this level of predictive power in living matter? And I agree, this is a tall order or something quite ambitious to want to do, but all I know is that in the one case that we worked on for 15 years in my PhD lab, we managed to do this. Okay, so again, we're taking a detour from flies here to think about bacteria, specifically to think about the lac repressor that binds to the DNA and doesn't let polymerase bind to it. And in the context of this simple system, you could think of 3 different regulatory knobs. One is the binding energy of the repressor to the DNA, the number of repressors, and also the number of promoters. It's not the same to have 100 repressors on one promoter to have one repressor on 100 promoters. Okay, so you can derive models based on the Coulombian statistical mechanics that predict the fold change in gene expression as a function of the number of repressors, and each curve here corresponds to different settings of these knobs that I'm showing you here, okay? So the point I'm trying to make is that these are 0 parameter fits. These curves existed before we took the data. And then we went on and took the data, and what we showed is that to a very large extent, you can actually predict the level of gene expression from knowledge of these little parameters, okay? Without any other free parameters, right? Again, 0 parameter fits there. You were either right or wrong. Not only that, note that all these curves look different, even though they all stem from the same fundamental, the same phenomenon. And the reason for that is that we didn't plot this as a function of the natural variable, okay? And you can show that if instead of the repressor copy number, you plot the repressor fugacity, which is something equivalent to trying to map a non-ideal gas onto an ideal gas, all the data collapses onto one master curve, okay? So when people give me a hard time saying biology cannot be predictive, I show them this and say, I don't know, like, in the one case we tried hard, it works. So like, but then of course you could say, well, okay, this was for bacteria. Bacteria is simpler. Eukaryotes have a lot more bells and whistles. It's going to be more complicated, and I agree 100%. Eukaryotes, animals have a lot more bells and whistles. The question is not, but then it's not like you want to be paralyzed in fear, but the question is which ones of these molecular parameters, what are the details that you need to know in order to reach this sort of understanding? Is there a question in the chat? Or is that for me?
Sure. There. Yeah.
Felix, do you want to just ask a question?
Yeah.
Can you?
During the tutorial, you're very encouraged to just turn on your mic and ask questions. So Felix, I think, who was asking the question.
Anyway, so I guess I can read the question. There you go, Felix. Go ahead.
I did not want to interrupt. Thank you.
No. But please interrupt. It's all good.
In cellular processes, often the number of molecules involved is very low, so you maybe would need to expect stochastic effects to play a role. Like in the example with the electronic circuit, also if you have very low currents, you also see stochastic effects that you cannot calculate so easy, maybe, right?
Absolutely. So in the bacterial case, I can tell you this is all averages, so we're averaging the noise. Though work from my PhD lab has shown that you can even predict the noise in gene expression as a function of the repressor copy number and as a function of the binding affinity of the repressor to the DNA. Okay, where basically you assume a two-state model like the one that I talked about, and I'm gonna show a little bit more. So yes, stochastic effects are there, and I think they're interesting from two perspectives. One is how do cells buffer them, but also they provide yet another lens through which to understand what's going on at the molecular level. And bursting is one example, right? Like you got bursting in principle that makes it more stochastic than if you had a constant level of transcription.
But you don't think that they are so important that they might break down this deterministic description?
I don't know. I mean, in this case, it seems to like we seem to have been able to average them out and still get predictive power, right? Whether that's going to be true in the fly, I think the jury's still out there. I can tell you that every time we try to use these thermodynamic models or statmic models on fly development, we fail.
Okay.
So, but that could be that it's very noisy, but also could be that the system is out of equilibrium. My intuition is that it's because the system is driven out of equilibrium more than anything else. But I think the jury's still out there. So I can't claim the sort of victory on the fly front just yet.
Okay, thank you.
Sure. And again, please just stop me if you have any questions. All right, so I'm gonna maybe put the chat window here. There we go. All right, so so as I said, maybe like you got to figure out what the molecular details are that you need to keep, that you want to keep in order to try to write your equations, and that's why I was advocating for maybe zooming in a little bit more into these input-output functions to not only worry about how much activator or repressors are in the cell, but actually try to figure out how much is bound. Not only worry about the output, the average output level of expression, but actually figure out what's going on in single cells. And this is particularly important in development where everything is happening very fast, right? In the bacterial example I showed you before, everything is in steady state. Exponential growth, you're relatively happy, like ignoring the time component. But here you gotta really acknowledge the passage of time and what each individual cell is doing. And I gave you the speech about the different types of microscopes. I find it funny, but it's kind of an interesting way to approach the problem. I think or alternatively, kind of following up on Darwin's idea of having a sixth sense when you try to address problems from the standpoint of mathematics. Okay, so what I want to do is go into a little bit more detail into the first two parts of the talk that I also mentioned during the 50-minute talk, and then go into new types of microscopes once again that we're trying to develop in order to solve this little conundrum of how do you reconcile binding of transcription factors and transcriptional testing. Okay, so once again, we're going to be looking at the fruit fly. It's regulated by these 4 transcription factors, 2 activators and 2 repressors. The even skip strike 2 is regulated by these 2 activators and repressors. And we are using this MS2 technology. I don't know if anybody has questions about it, but feel free to ask them now. But just as a reminder, each one of these dots tells you how many polymerases are actively transcribing an individual gene as a result of the individual decisions of a promoter to either transcribe or not. Okay, and as I showed you, when you zoom in, you get this sort of dynamics of the number of polymerase molecules actively transcribing as a function of time that looks like trapezoids, and this can be related to the rate of polymerase loading through a very simple model where the promoter is on transiently. Okay, so these trapezoids indicate a transient turning on of the promoter. Okay, and as I mentioned, and Tineke also reminded us, people have seen these bursts all over the place. Like Ido Golding has seen them in bacteria, Dan and Tineke and many others have seen them in yeast. People like Rob Singer have seen them in mammalians, and it's pretty interesting. I mean, Dan Larson has some examples of genes that seem to be more consistent with a single one-state promoter, and there are some examples in bacteria as well. But for the most part, when people look, they seem to see bursts. Okay, which, it's interesting. People like Heinrich Berger, if you don't know him, he's at Santa Cruz, UC Santa Cruz, and he claims that there might be some meaning in having these bursts, that actually they allow you to filter some other types of nodes. So there are interesting ideas out there that maybe bursting is not just a nuisance, and maybe it's being useful. Okay, and Gaspar Tacek has also some interesting models related to this. And as I mentioned, the simplest way that you can understand this is through this 2-state model. Okay, where you can be either on or off. You could imagine more states in this model, and people have been thinking about that. How you figure out how many states there are. This goes very much back to Clelia's talk in terms of model selection. It's a pretty tricky problem, in my opinion. But this is enough to capture the phenomenology, this 2-state model. And once again, you can define burst frequencies, burst duration, and burst amplitude.
I did have a question about the 2-state approximation. So you've already made the point that it's probably a phenomenologically acceptable approximation, but it could be more complex, right? And that in some sense must be the case, right? Given how many molecules and ultimately atoms are involved. But I wondered, if at the level of temporal statistics, you have support for this 2-state picture. I mean, this 2-state picture where you just have one time constant per state.
Yeah,
right?
that's a great question.
You should be able to look at.
Yeah. So we're just starting to, with these hidden Markov models that I'm going to talk a little bit more in detail in a second. You can, in principle, get these waiting time distributions. Yeah. We're not entirely sure that we trust them just yet, because you can. It's easy to get average parameters, but now whether you can get the distributions, well, that's a different question. I can tell you that if you believe them, I can't remember. One of them seems exponentially distributed, but the other one doesn't.
Okay.
But I'm not believing it 100% just yet.
Yeah.
But I agree. There's gotta be interesting information in the waiting time distributions.
Yes, so that would be exciting too.
Yeah, I agree 100%. Yeah. And yes, so I'm very interested in using the distributions as yet another microscope or a lens through which to dissect the biochemistry that is going on in there.
Okay.
Yeah. But I mean, people believe that, Alex Walchuk has some examples. She believes that one of these promoters has 3 states. Jonathan Jupp has a model of a continuum of states of bursting in actin. So people are very much thinking about this, but I think we're still struggling with coming up with a good model selection thing. I can tell you that for our hidden Markov models, I could tell you, depending on which model selection criterion I chose, I could pick the answer that I wanted. unfortunately. So we haven't been able to figure that out ourselves.
Can I ask something about the use of the hidden Markov model?
No, say it again. Can you start over?
Sorry, I wanted to ask a question about the use of the hidden Markov model. Is there any risk that by choosing the model that you kind of influence the distribution of the on and off rate of the on and off time of the binding times towards kind of the model you selected? Or is
I think there is. I think you're absolutely right. Yeah. I think, and that's where the challenge of model selection comes in. And I don't know myself how to solve this well just yet. Yeah. So I agree 100%. So there's such a thing as biasing your results because of that. Yeah.
Thanks.
Yeah. So, but before even the hidden Markov models, I mean, think of the fact that, this mean rate of transcription, or which tells you how much there is produced on average, has to be the result of modulating one of these bursting parameters, right? And as I mentioned, you can in principle do it by any combination of these three strategies, by modulating K on, K off, and R. Okay, and so a fair question is to ask how much do each one of these parameters changes as a function of time. By the way, we're very obsessed with bursting. I'm talking about the whole field, but it turns out that the modulation of the mean transcription rate due to bursting is only accounts for a small fraction of the stripe. Most of the stripe is created by deciding when to start bursting and when to stop bursting altogether, by basically regulating the transcriptional time window. And I'm happy to talk about that later. But nevertheless, I think bursting is an interesting window into which to try to understand what's going on at the biochemical level with what transcription factors are doing. So once again, we got to do some sort of inference. And for those that are not familiar with these hidden Markov models, like basically what we're trying to do is predict the hidden promoter state, right? Something I cannot see from the observable fluorescence. And if you're like a ion channel aficionado, you might know of hidden Markov models in the context of predicting or inferring the state of a channel, whether it's open or closed, given an observable current. And the big challenge, the reason why we were stuck sitting on terabytes of data without being able to analyze them, is that these hidden Markov models are usually designed for systems where there's a one-to-one correspondence between the state of the promoter, the state of the channel, and the current that you measure. Okay. However, in the MS2 case and the transcriptional case, the same promoter state can lead to two different fluorescence values. There's a memory. It doesn't mean that the promoter is not Markovian, but the observable has a memory. And that comes from this memory kernel, if you want to think about it, of how long polymerase stays on the gene. Okay, so that's where the memory is coming from. So what Chris, Vache, and Nick figure out is basically a compound state hidden Markov model, where just like when you're playing chess and you need to, the computer needs to figure out all the different combinations of moves that you can have ahead, and that grows exponentially. What you do is you go back in time and you ask, given an observable fluorescence right now, what is the most likely progression of promoter states that over the memory time window, which is given by how long the polymerase stays on, what is the most likely progression of states that can give me the observable fluorescence? Okay, so that's basically what it's doing. It's basically doing hidden Markov model, but on a memory time window instead of an individual state, like in an ion channel. Okay, and this allows you to do this inference. And it takes a few hours in a cluster. I mean, it's not trivial, but it works very well. And people like what's her name, Hilary Ash at Manchester has figured out a way to be smart about this and even go a little bit faster. And so the result is this very cool thing, at least in my opinion, but I guess if I don't find it cool, I wouldn't be talking to you about it, that allows us now to predict the most likely state of the promoter as a function of time, as opposed to looking at these fluorescence traces. Okay, and I think this is quite an enabling technology, because until now, what people were doing is either set a threshold and say, well, whatever's over here is on and whatever's over here is off, or break the promoter such that the bursts would be so separated that you would see trapezoids independently. So this allows you to look at the bursting dynamics in an endogenous setting. And once again, it's just fun to see these nuclei bursting, even when you're at the highest level of expression right in the middle. Okay. All right. So I showed you how we can measure these different bursting parameters as a function of position, and we see that their burst frequency is modulated, the burst duration stays the same, and the burst amplitude also changes. Okay. And as I said, this is kind of consistent with what people have seen throughout. Like most of the time people see control of burst frequency, but more and more people are starting to see control of burst amplitude, also sometimes control or burst duration. Okay. And so the question is, one of the first questions is, how general is this? Okay. And one of the cool things that I didn't tell you about, or that I hinted at, is that this even skip gene actually doesn't produce just one stripe, it produces 7 stripes. And it produces 7 stripes by using 5 different enhancers that have different sequences that respond to different transcription factors. Okay. So now you can repeat the analysis in all of these 7 stripes. Okay, so you can see them here, 1, 2, 3, 4, 5, 6, 7. Okay, and what you can do is just run these movies through our hidden Markov inference algorithm and plot burst frequency, burst duration, or 1 over burst duration and burst amplitude as a function of position. And what you see is that it wasn't just for stripe 2, all the stripes seem to be created through the same qualitative mechanism of upregulating burst frequency in the middle, leaving the burst duration basically the same, and upregulating burst amplitude in the middle of the stripes. Okay, again, this is kind of weird. Like these are different transcription factors that are regulating each one of these stripes, yet they seem to have the same qualitative. They seem to be using the same qualitative strategy to generate the stripes. What's even more striking is that this agreement is not just qualitative; it's also quantitative. Okay. So this goes back to being able to plot parameters in terms of natural variables. Here I'm showing the bursting parameters as a function of the position along the embryo. But you can plot them as a function of the mean transcription rate of each cell. Okay, and what you can see here is that here I'm color coding the lines with respect to which stripe it is. And what you can see is that they all fall on top of each other. So what I'm telling you is that quantitatively, all the cells are bursting in the same manner. Okay, or their control is implemented in the same manner. Basically, if I give you a cell that is bursting from one stripe, you have no way of telling me or to figure out which stripe it came from, because they all look the same. And this is weird, at least to some degree. You could imagine, well, maybe there's some biochemical constraint that forces you to only actuate bursting through given biochemical pathways that have to do with controlling K-on and the rate amplitude. Or alternatively, you could imagine that maybe there's some evolutionary advantage. Maybe there's there's something good about bursting, and there's something good about bursting with these particular bursting parameters. The problem is that this unity, there seems to be unity in some in this set of genes, and there seems to be only unity in all the genes, but the unity is different. Okay, so this is work from Thomas Craig using single molecule fish, not using hidden Markov models. They can do really high precision measurements of distributions. Here they measure all the gap genes, all the genes that are in the layer above the stripes, and they measure K-on and K-off. They also measure R. I'm not showing it to you, it's constant. Okay, so they show that both the burst frequency and the burst duration are modulated. But even more interestingly, they saw that there's a conserved quantity, that K-on plus K-off is a constant. Okay, so it's not like the system modulates K-on and K-off. It modulates something that dictates K-on and K-off. You kind of modulate the two independently. Okay, so then again, there's a unity here, but it's a different type of unity than the one that we're seeing in the stripes that I just showed you before. So I don't know. I'm confused, and if you're not, I would love to understand why you're not confused. But basically, this brings me back to the idea that clearly we're missing. We've gotten really good at figuring out which bursting parameters are being regulated, but we still don't know how they're being regulated. And that's where this concept of, well, let's try to understand what's going on at the level of individual transcription factors binding to the DNA and dictating bursting dynamics. Okay. And I put forth these there's two scenarios. I mean, they're not mutually exclusive, and there are even more. but you could imagine that sometimes transcription factors can have to be on the DNA for the whole duration of a burst. Maybe they just need to prime the burst, and then they can go do their bidding somewhere else. So my point is that this calls from some sort of correlation between binding and transcriptional dynamics. Okay. So any questions so far?
If I could ask a question to confirm.
Yeah.
You had shown, I think in 2 slides ago, that the off rate didn't seem to be modulated so much, but in this slide now you're saying it is modulated.
Well, yes, that's a confusion. In the stripes, in the 7 stripes, right? These 7 stripes, they none of them is created by modulating the off rate.
Okay.
Okay. But now you look at a different set of genes, the gap genes.
Okay. Sorry. Yeah.
Exactly. And now K-on and K-off are modulated, but in a way where they're not independent.
Yeah, that's really puzzling.
It is. Well, but I mean, it goes back to, do we have the right model? And clearly we don't, right? So the question is, what is the biochemical model, the set of equations that you could imagine that would give you some control parameter that dictates these effective parameters of K-on and K-off in this fashion?
Yeah. Yeah, it's very
right. Yeah. So I don't. I mean, I don't really know what's happening, but it's cool, and you can imagine different things like, if you deliver a package of transcription factors and they need to be exhausted, maybe you can make up stories about it, but also we need to figure out how to test those stories. Okay.
And maybe one follow-up question on the same issue. So the data of Thomas Kreger, that was not generated using your procedure, right?
No, it was generated using single molecule fish.
Yeah.
They used some very cool thing of labeling both sides of the gene, 5' and 3' with different colors, and some fancy MC type of approach to try to find the most likely model.
But of course, it would be very interesting to sort of use both procedures, apply both techniques.
Absolutely. Yeah. Absolutely. And this is the problem with biology, right? Like everybody does their own thing and they never try to do the advantage of Thomas's approach is that he can do it on the endogenous genes. Like these genes have not been modified in any way. We are now making, and I think Thomas has some of these too, we have now made CRISPR fusions to hunchback giant crabs and I think even cripple. And we're trying to measure them using our MC approach and see whether we see the same thing. Yeah. And in fact, one of my students, and this is something also very, very interesting to think about, is one of my students is very curious about the temperature dependence of these parameters. So we're starting to do these experiments in controlling the temperature between, like, I can't remember, maybe 18 and 28 degrees or something. Like, like just try to find knobs where you can start like dissecting what's going on under the hood. Yeah, and so I think Thomas's data is great. Obviously, it assumes things about steady state and such because they have to. So it would be great to actually measure the same thing with this, with 2 different methods.
Yeah, thanks.
Sure. Any other questions about this? No? All right. Okay, so I mentioned, hey, like a simplest model is one where your activator binds to the DNA, and when it binds, it turns the promoter on, right? And this makes the prediction that burst frequency has to be equal to the activator rate, that burst duration has to be equal to the activator residence time. And so we went on to measure this, okay? And I talked to you about the latest light sheet. If you want to ask more questions, just let me know. But basically, if you're familiar with turf, this is like turf, but in volume. It's truly fantastic, but really hard to build and operate. And once again, what you see here are these little flashes that correspond to BCOY GFP binding events onto the DNA. And we can measure the waiting time distribution. Marvin gave us a little bit of word of warning, is this really that exponential? But, on average, what we see is 1 second. And, but the bursts last 5 minutes. I have to caution you, and Tineke reminded us of that she doesn't see in yeast that big of a disagreement. Okay, but the bursts in yeast are very short compared to what we see here, to what people see in early development, and even in some somatic tissue in mice. The bursts seem to last on the order of minutes. Okay, and anything you measure in terms of residence times, they never last more than 15 seconds or so. Okay, and And this is kind of reflected by this figure from our review, we just wrote, where you can see how there's a mismatch. Most of the time scales that you could imagine, like, the clusters of RNA polymers that form, the looping, pulsing, all of that, it seems to take too long compared to everything, to the bursts that we can see. Okay. So what we decided to do is try to come up with a slightly more sophisticated model. And this goes back to Marvin's question about, what is the binding affinity and all of that. So now what we're going to do is adopt a fully kinetic model where we have multiple binding sites. Here I'm showing you 3, but I think the results I'm going to show you are for 6 binding sites. We're going to posit that the number of RNAs made per unit time is proportional to how many activators you have bound. And there's good reason to believe that there's some linearity between number of binding sites and expression rate. And we're also going to assume that all the binding affinity, all the binding of constants rates are the same for all the binding sites. They're all identical. Okay, just to keep it simple. We're going to impose the binding dynamics that we measure in the lattice slide sheet. Okay, and ask, what does the transcription rate look like in this case? So when you simulate the transcription rate as a function of time, what you see is that the rate of transcription is unimodal. There are no bursts here. Okay, there's no on and off. The reason for this is that each transcription factor just binds there for a second. So when we reason, and this is not very crazy given years and years of molecular biology, is that maybe there's some game that the transcription factors are playing when they're both together. Basically, but maybe there's some cooperativity that is stabilizing the molecules on the DNA. Okay, you can imagine also cooperativity that catalyzes the binding, but this is the most classic type of cooperativity. The results are the same. When you include this sort of protein-protein interaction, now you can get bursting. Once again, we kept the binding and unbinding of individual molecules the same as in the lattice slide sheet, and we put some reasonable interaction energy of like 5 kT or something like that. Okay, and what you get is not only bursting, but also bursting on the right timescales. Okay, so again, this is not saying that the model is right, but it's just interesting that you just gotta add that little extra element to get bursting out of this simple model. Okay, but now you might ask, what's happening? How do you reconcile this idea of short binding events with long bursts? Okay, so what you can do, fortunately, in a simulation is actually measure everything with infinite precision. So what I'm showing you here is the dwell time of each one of the events that we had in our simulation. Okay, and what you see is that there are 2 regimes. There's a short binding regime this is what we can capture with the lattice slide sheet, for example, because GFP bleaches. Okay, and in this regime, the distribution looks exponential, like this little black curve here, but then you have a bunch of long-lived events that are clearly not exponential. Okay, and you could wonder whether these long-lived events are the ones that are actually driving bursting. So the cool thing is that you can do the deletion experiment in the computer. This is the dynamics that we get if we have both short and long events. And what we did is we deleted the long events. Like basically every time a transcription factor was bound for more than 2 seconds, sorry, we got rid of it. And what you see is that bursting disappears. So what this shows you is that the long binding events are necessary for bursting. Okay, at least in the context of our model. And so once again, the question is, why don't we see these long binding events? Okay, and as I mentioned, one reason is that there's bleaching, and we need better fluorophores, we need better technology. Although I think there's only so many photons that you can get out of a fluorescent protein. But I think the main reason is that we've been staring at random locations, like you're going to capture some specific sites, some non-specific sites. That's all good. But ultimately, you want to look at the binding at the site of nascent transcript formation that you're looking at. Okay. So how do you do this? All right. So let me just remind you that there's no amount of super resolution microscopy that is going to allow you to figure out whether a transcription factor is bound to a specific binding site. Okay. At least I don't know any of it in fixed tissue, and let alone in living embryos. Okay. So what we're trying to do is to come up with some more nuanced ways of measuring binding based on some really cool work by Junma and Ido Golding. So here, what they try to look at is bicoid binding to hunchback. So bicoid is an activator, hunchback is one of its targets. They looked at bicoid molecules at some random location in the nucleus and at bicoid molecules in the vicinity of the hunchback site of nascent transcript formation. And what they show is that there's more B-code in the vicinity of hunchback than at a random site. In fact, there's a non-trivial dependence of the number of B-code molecules at the locus as a function of the B-code concentration, which they claim, although this is kind of strange, but that these are binding curves that they're measuring. Okay, so this is fantastic. But once again, this is in a snapshot. So could you do this in real time? Okay, so we set off to do this using a slightly different system than the stripe that I was showing you, which is the dorsal activator activating SNL. So the details don't matter, but basically dorsal is expressing a gradient now along the dorsal ventral axis of the embryo and leads to the expression of SNL in a domain. Okay, and you can play the same games with our MS2 technology, and what you see is that SNL is expressed in this broad domain that you see on the ventral side of the embryo on the bottom. Okay, so what we do is we simultaneously image MS2, SNL MS2, and the concentration of dorsal fused to venous. Okay, so green is the input and magenta is the output. Okay, and then we ask, if I center my window around a transcription spot, do I see more dorsal around the transcription spot than somewhere else? And the answer is no. Right? It's super noisy. There's nothing there. No matter where you look, if you take a snapshot, you don't see anything. The cool thing is that once you start averaging these frames, you only need 50 snapshots, 50 time points, to see that there's an enrichment of dorsal in the vicinity of its SNL target locus. Okay, so we believe that we're measuring a signature that reports on binding in a living embryo. And you can start doing controls. This binding or the signal seems to be related to the regulatory function, because this is dorsal at its target SNL. This is dorsal at its non-target hunchback. Okay, this is picoid at its target hunchback, and this is picoid at its non-target SNL. So basically, dorsal target SNL, we see it enriched there, but nowhere else. Picoid targets hunchback, we see it enriched there, but nowhere else. Okay, so this is all good, and this is basically what people could do with fixed tissue. But now we have an extra dimension, which is the dynamics. Okay, so this is dorsal, the transcription factor we're looking at, enrichment as a function of time at a control locus that it doesn't interact with. This is, yep.
Just quickly ask. So that averaging, was it done over time or over many spots?
Either way, you can do it over time, over many spots. These ones are over everything we have.
Yeah.
Both time and spots. Yeah.
So I mean, maybe just to relate it back to the very short bindings. I mean, if you had done the lattice light sheet experiment in a way that you just integrate over much longer times effectively, so that your exposure time with light sheet, I don't know exactly, what an exposure time.
Well, you would have to, but you need to center it around like a locus, right? Remember, we're seeing in the light sheet, we're seeing binding events all over the place.
Yeah.
Here we are looking in principle at binding events at a locus.
But I guess if you were to just integrate over a very long time at very low intensity, wouldn't you see anything? I mean, your exposure time to
over a long time.
The sub-second binding and unbinding, I guess, is, exposure time is very short. But if you were just dilate the time in that experiment, I think
whether you would see this enrichment, or whether you would see dynamics.
No, the localization. Yeah,
you see it. You do see it, absolutely. And people have shown that. Our colleagues Mike and Xavier have seen that using the lattice light sheet. Yeah, absolutely. The lattice light sheet, we're working on reprogramming it because it's harder to track the transcription spot over the timescale of bursts.
Mm-hmm.
Like single molecule imaging requires very, very fast and short movies.
Yeah.
But because of the separation of timescales, looking at the MS2 spot requires longer movies.
Yeah.
So you got to come up with a better way of doing the tracking in the lattice light sheet to actually do truly single molecule detection while imaging transcription.
Okay.
Yeah.
All right. Thank you.
Yeah. Sure. Okay. So I was saying there's some dynamical signature to this enrichment. Maybe this is in hunchback, and this is like anecdotal almost, right? I'm telling you that if I look at SNL now, there are excursions
Okay, and presumably these excursions are what increases the average occupancy of dorsal SNL. Okay, so the question is, do these fluctuations correspond to binding events? And if so, are we capturing these long binding events because they're on the right time scale? And the second question is, do they correlate to bursting? Right. So if you could correlate these fluctuations, like we know that enrichment is related to binding, but if you could correlate these fluctuations with bursting, now you're starting to get a window into what transcription factors are doing at the promoter. Okay, and these are very early days, but we think we have a way to do this. And if you stare at this, clearly the signal sucks, right? It's going to be really hard to know something about an individual burst. But the cool thing is that you can invoke basically the same type of logic that leads to super resolution microscopy, or more related to this in neuroscience spike triggered averaging, to try to combine information from multiple bursts to spread out the information over time so you can increase your sequence size. What do I mean by that? On the top, I have the output transcriptional activity. On the bottom, I have the dorsal enrichment, the input enrichment of the transcription factor. Fortunately, the output transcriptional activity from MS2 can be decoded using the Hiro-Markov models. And now I can start aligning all the bursts with respect to their start and also the signal from the protein. Okay. And, we think we're starting to see a signal, though we also think we need to switch to the lattice light sheet. This is the signal to noise is too crappy it's like almost there. Okay, so I think this might work, and that's why I'm very excited about this type of experiment, because if you collapse now these 3D plots, that's where I think you might be able to reveal the so-called anatomy of a burst. You might be able to say, given a burst, when is the transcription factor there? You could imagine also tagging pioneer transcription factors and seeing whether they appear before, during, or after a burst. And you could even use these nice sensors that bind to specific histone modifications and even see what the histone landscape looks at the burst. So the point is that if we get this to work, we might be able to reveal the biochemical sequence of events at high spatiotemporal resolution at an individual locus in a living embryo. Okay, and hopefully this will give us the level of detail that we need to actually start putting together these predictive models of cellular decision making. How many states there are, what are the transcription factors actually doing when they get to the promoter. Okay, so I'm gonna end now, and as I've been following around with this idea of microscopes. There's this in vivo microscope to reveal transcription with MS2, this computational microscope to infer promoter state, this theoretical microscope to make predictions about what transcription factors are doing when they're bound, which led to testing the model using lattice light sheet microscopy, which led to a completely to some crisis, I would say, in the theory, and which led us now to try to develop this data analysis microscope, which we call burst trigger averaging, and which we hope will be able to tell us about what the transcription factors are doing during the burst. Okay, and I mentioned this already. I don't need to say this again. But I guess I need to acknowledge these guys again. Fantastic group of people, my lab, and everybody for your attention. And I think we still have a few minutes maybe to answer some questions. So yeah, thank you very much for having me. It's been a great morning. And the fun thing is that now I still have my whole day to do stuff. So
great, wonderful.
Yeah, thanks.
Beautiful talk.
Absolutely.
Yeah, so there's already been several questions, but I would encourage people in the audience to still jump in with a few more. As Hernan said, his day is just starting.
Yeah, I could start maybe.
Yeah, sure.
Oh. Would you like me to start?
Yes, please do.
Yeah, so thanks for a great talk again. Beautiful. So I just have a question on this very last part that you mentioned, where you basically try to correlate the promoter state with the subsequent transcription, right? Exactly this slide.
Yeah.
And so how does this procedure go in a little bit more detail, and how would it compare to a much more brute force sort of getting statistics on the 2-point cross-correlation function between, let's say, the promoter state, which you extract from your hidden Markov model on the one hand, and on the other hand, let's say, the dorsal activity. So you just do a 2-point cross-correlation function.
Yeah, we've done those cross correlations as well. Yeah, and there seems to be a correlation. What else can I say? The peak of the correlation seems to be centered basically around 0, which is interesting. So they seem to go together. But it makes it harder to break this apart into, let's say, if I have enough data, now I could ask, what happens if I only average bursts of a given length? And what happens to the signal during that? So there seem to be some correlations, though I gotta say, this has been really hard because it's one of those things where you write long list of artifacts, and we've been going through each one of them. And as we get rid of artifacts, we keep losing signal. And so right now, that's why we need to go. We need to go to fancier microscopy than confocal microscopy to do this. But yeah, so the 2-point correlation function has been useful in telling us that there is something there, and we're thinking of how to use it well. Though I think the nice advantage of this is that it allows you to parse the data into more sophisticated things. For example, we think that dorsal is controlling burst amplitude mostly. So then I could try to only grab bursts of a certain amplitude range and ask, what is dorsal doing there? Like, is amplitude correlated with how much dorsal is being delivered to the locus? And with these cross correlations, I think you're going to lose probably the information about the absolute intensity.
Yeah, it's very true.
But they're useful nevertheless. And actually, they're a good way to develop intuition and to make sure that you're not fooling yourself, right? Because they should be a signature there.
Thanks.
Sure.
Okay. Other questions? I might ask one about, I was very curious how you set up the model with the cooperativity that produced evidently a better match with the timescales, right? So there's multiple ways of introducing, kinetics into these cooperative models, which often are just described in equilibrium. So I was curious how.
So I mean, we try both ways basically. I mean, these are all pairwise interactions. These are not higher order interactions, higher order cooperativity, which could also save the day. But this particular example that I showed was one of stabilization of the unbinding. Okay, this is more the classic protein-protein interaction, lambda repressor type of thing. Okay, you get the exact same result basically if you put the omega on the binding. Okay, such that the presence of one activator catalyzes the binding of the second activator.
Okay, sorry, that was already in the slide. So omega is the cooperativity coefficient.
Omega is the cooperativity,
and that's basically scaling down the unbinding.
Exactly, but again, you can put multiply binding by omega, and you get the same answer basically.
Okay, yeah, I see. Yeah, that's interesting. So you would kind of just expect an exponential decrease in the time scales with the number.
Yes.
I see.
Yeah, and so that traps you effectively in the high and low states.
I see.
That's why this is happening, because you can see it here, right? It stays here, but it's very unlikely, to go back for it to get out of here, and the same thing down here.
Yeah, and
so there's. Go ahead.
Do you understand why, in the end, it ends up with a distribution, dwell-time distribution that looks like kind of like a power law in the? I guess
no, that we haven't explored in detail yet. No, you mean this?
Yeah.
No. That we haven't explored in detail just yet.
That was a numerical result, I guess.
This is a numerical result. Yeah. But I mean, it is an interesting one, and we should explore it. Yeah. We should see what we can see. Yeah, absolutely. I mean, I was more focused on, are there long-lived events to begin with, right? Because ultimately, the answer, what this model is telling you, is that there are indeed long-lived events, even though what we can see are the short-lived ones.
Okay. Thank you.
All right.
I might have a very naive and simple question. Right, so in eukaryotic transcription, you often have this assembly of these generic factors, transcription initiation complex. So does that play any role here? So what's known about that?
I wish I knew something about what happens with them at these timescales.
Yeah.
Because I don't know if the whole machinery needs to be reassembled with every burst.
Exactly.
I don't know. Yeah, I really don't know. I mean, same thing about nucleosome positioning and all of that. Like, all the information we have comes from either biochemical experiments that are pretty hard to reconstitute, or from genome-wide experiments that they just don't have the spatiotemporal resolution.
Yeah, exactly.
And that's why I'm so excited about this idea of the anatomy of a burst. Like, maybe it gives you the resolution, although it's average over many bursts, maybe it gives you the resolution to start answering these questions on that timescale.
Yeah, I think it's very interesting, right? Because now we basically have been, making a lot of progress with models that are essentially coming from bacteria, right?
Yeah. And so we have one paper, for example, showing that there's no way you can explain the initiation dynamics, so the time it takes after mitosis to turn on transcription, using thermodynamic models. So, and that instead you need to propose a model where you need to go through different irreversible kinetic steps before you can start transcribing. Now, now you could say, well, it's maybe the unwrapping of a nucleosome. But yeah, I can tell you, every time we try to reconcile these thermodynamic models, they fail, which is an exciting thing. And so we'll have to see what energy and all, what those things are doing here. But not only that, also, how do you test those models? Because this is not like the stuff where, like, really cool stuff by Joe Howard and others, where you can actually see the flow through the space. Like, you can see the flow of energy in some sense, because we don't have access to the microstate. So we need to come up with smart predictions about how that are distinguished between the different scenarios.
No, I agree. Yep.
Okay.
All right.
Yeah. So Max has his hand up. I don't know if that was just left up from earlier.
I think it was left up, but I have one question, I think, about this slide. So here it gives you this probability, which I'm not entirely sure how to interpret, but if you have the amounts of bursts you see, gives you that kind of a number for how many I don't know if this was bicoid, but for how many transcriptor molecules you need to achieve that amount of bursting.
I see. Like, how often would you get these binding events?
Yeah, I don't know exactly what the probability means.
Yeah. Here we didn't think about the concentration dependence, because we kind of plugged it into we didn't play with the concentration actually much. I think we put some reasonable values of concentration though, but we didn't try to look at the concentration dependence. So I would have to go back and figure that out. So what you're saying is that, sure, there's long-lived events, but are there enough of them to give you bursting?
Yeah.
Is that the question?
Yeah, does it fit with kind of your expectation of the amount of transcriptor that is there?
Well, I think they do, in the sense that, the timescales of these, this is qualitative, right? But the timescale of these bursts are consistent with what we see. But we haven't looked at this in a lot of detail, I have to say.
Good, thanks.
Sure.
Okay. Great.
Well, thanks, everybody.
Okay, well, I think, we have gone over time by about 10 minutes. But yeah, I think, it was well worth it. It was really cool stuff you're doing, Hernan, and the newer data.
Thanks, everybody.
Awesome. I'd be really excited to see what you learned from the temporal statistics that you're doing as well. So that's on the cards.
All right.